Hello, this is Cynthia Challoner, editor of the Pharmaceutical Sciences Manufacturing and Marketplace Report, a specialized biweekly e-newsletter of pharmaceutical technology and Biopharm International that covers technical, scientific, and market developments in the pharmaceutical manufacturing area. I'm here today with Annelies Onrait, Global Market Manager for Vaccines with Pall Life Sciences, to discuss advances in conjugate vaccine manufacturing. Welcome, Annelies.
Hello, Cynthia.
So can you tell me what is driving interest in conjugate vaccine manufacturing?
Yes, well, actually I would say there's two main drivers here. On the first hand, you have the several emerging markets manufacturers that are developing low-cost vaccines for local supply and that are looking to produce conjugated polysaccharide pediatric vaccines like HIV or pneumococcal vaccines. And on the other side, you have the big and small vaccine companies in the developed world who are trying to address new vaccine targets, and they are more difficult to achieve with traditional vaccine antigen technologies. So they're looking for more sophisticated approaches to get a good and long-lasting immune response, and these are currently in R&D, and some of them include conjugation. It's an approach currently in clinical phase development for Staph aureus vaccines.
Can you describe then a typical conjugate vaccine manufacturing process and what are the key technologies that are involved?
Because in a typical conjugated vaccine production, you're conjugating chemically a protein and a polysaccharide, you will first need a protein production and independently of that a polysaccharide production. So for both protein and polysaccharide productions, the starting point is most often a bacterial fermentation, where then the bacteria of choice will be selected to contain or produce high amounts of the protein or polysaccharide of interest. Then what follows is a purification process for either the protein or the polysaccharide, with the main objective here being to remove unwanted contaminants, such as the host bacteria or contaminating bacteria, other metabolites, including endotoxins, and some remaining media components. And this is done by a sequence of direct flow filtration steps, tangential flow filtration steps, chromatography, and precipitation. Toxic proteins like diphthery or tetanus toxoid proteins will then need to be inactivated, and this is done in a kind of hold step in the presence of formaldehyde. And then polysaccharide production especially can be some kind of trickiness because these polysaccharides sometimes are very long and they will then be chopped into pieces. And the further size distribution of the polysaccharides is relevant for the conjugation process and the quality of the vaccine. So size-based separations using several molecular weight cutoff tangential flow filtration membranes and size exclusion chromatography will be needed. Then for a conjugated vaccine, the most critical step is the actual conjugation, and this is the chemical reaction. So the proteins and the polysaccharides will be made reactive. They will be reacted together using a number of chemicals, reagents, and the efficiency of this step especially is determining for the yield of the conjugated vaccine, and it's also the main part of research currently. So then the unreacted species, they will be removed using the size difference between a conjugated and a non-conjugated compound. So for example, they can be flushed out using a tangential flow filtration membrane with a larger molecular weight cutoff. And then the conjugated product is going to be sterile filtered and then going into final formulation and sterile filling. Most conjugated vaccines are multivalent vaccines, and this means that the final vaccine will consist of multiple different conjugates, typically with varying polysaccharides. So each of the conjugates will be produced separately, and then they will be mixed together just before the filling. So the process of fill and finish of such vaccines can be logistically very challenging, and because of that, the single-use fill and finish operations have been very appreciated and widely adopted now in conjugate vaccine manufacturing. So there, the biocontainers with the sterile filter single valency conjugates are going to be connected to pre-sterilized tubing manifolds using sterile connectors. This allows them to aseptically connect multiple valencies and pool them into a single mixture in a closed sterile system that reduces the risk of external contamination and cross-contamination from the prior batches or cleaning residues.
Given the existing process for conjugate vaccine manufacturing, what do you think are the unmet needs with respect to equipment and processing that are driving the development of new technologies?
So the main limitations in conjugate vaccine manufacturing come from the overall complexity, which is due to these multivalent combination vaccines, and the associated cost of this manufacturing. And it's also there's also this increased regulatory push into a better characterized and a more pure product. So this latter especially emphasizes the need for better assays in the process and for QC tests. So the molecular side of the polysaccharides is going to be important for the efficiency of the conjugation. So the possibility to better purify these fractions with chromatography and tangential flow filtration will increase the yield. There's also some polysaccharides that can get viscous, which can complicate the purification process, and it will require further optimization. Now, some conjugated molecules can become reasonably large, and therefore they will require an optimized sterile filtration approach or even a complete aseptic processing. So then use of single-use technologies are going to be really indispensable.
Can you give some specific examples of recent advances in conjugate vaccine manufacturing equipment and process technologies that have been developed in response to these drivers, and what benefits do they provide?
Yeah, so a first big improvement in polysaccharide production came by moving away from solvent precipitation into a chromatography-driven purification process, which resulted in a more robust and a better controlled process and a higher purity product. Single-use technologies that have been combined with automated single-use systems have helped to run complete processes aseptically or to simplify the finish and fill operations. So single-use tech systems have reduced the risk of process contaminations to ensure product purity, and it also reduces the cost of the facility due to reduced clean room classification requirements, reduced WFI and CIP systems, and less capital expenditure for stainless tanks and pipings. Now, because of the chemicals that are used in the conjugation processes, extended compatibility information for the single-use plastic parts is required, and this is the kind of validation support that we are providing to our customers.
Then finally, what would you say are the key issues that remain unaddressed for conjugate vaccine manufacturing? Are any technologies in development that might overcome these limitations?
So cost and process simplification are the main issues that further need to be addressed with conjugate vaccine manufacturing. Single-use technologies are still relatively new to the field, especially in final formulation and filling. However, these issues are more related to the lack of familiarity, and they will resolve with more experience. The absence of fast analytical tools to measure polysaccharides, reacted polysaccharides, and conjugated polysaccharides is critical, and we are looking into how we can address this with our Forte Bioinstruments platform. But overall, the real optimizations will have to come from finding better conjugation technology with higher yields, either chemically or using new approaches such as in vivo conjugation, using E. coli, which is, as an example, that is now currently explored by some small companies such as Glycovaccine in Switzerland.
Well, thank you very much, Annelies. This is Cynthia Challoner, editor of the Pharmaceutical Sciences Manufacturing and Marketplace Report. I've been speaking with Annelies Sandreit, Global Market Manager for Vaccines with Pall Life Sciences, about conjugate vaccine manufacturing technology. Thank you for listening.